Well, welcome everyone to another seminar in the 2020 Monash Biomedical Imaging webinar series. I'm Associate Professor Michael Farrell and I'm going to be your host today. I wish to acknowledge the people of the Kulin Nations on whose land Monash University is positioned. I pay my respects to their elders, past and present. On a housekeeping matter, questions for our speaker can be submitted via the Q&A facility, which should be visible to you. And so answers to your questions will be provided at the conclusion of our speaker's presentation, unless of course Winnie decides she wants to tackle them along the way. In any event, it's now my honour to introduce our speaker. Winnie Orchard's a doctoral candidate at the Turner Institute of Brain and Mental Health and Monash Biomedical Imaging at Monash University. She's also a scholar with the Australian Research Council Centre for Excellence for Integrative Brain Function. Notably, Winnie plays a very active role within the community of postgraduate researchers more generally in her capacity as the Victorian representative on the Early Career Research Committee of the Centre for Integrative Brain Research. And Winnie's also distinguished herself in the level of research impact she's achieved so early in her career. Some of what you'll hear about today has already captured the public imagination. Media attention's a rare achievement for scientists, and Winnie's made a flying start in communicating her discoveries to a wider audience. And so, with no further ado, I will now hand over to Winnie to speak to us about the neuroscience of late life parenthood. Over to you, Winnie.
Thank you, Michael. I'll just share my screen. Is that working? Can you see?
Yes, that's
yes.
Fine. Thank you, Winnie.
Beautiful. All right. Before I get started, I'd also like to take this opportunity to acknowledge the traditional custodians of the land upon which I live and work as the land of the peoples of the Kulin Nations. I acknowledge their elders past, present, and emerging. I also pay respects to that. I also acknowledge the Aboriginal and Torres Strait Islanders listening here today, also paying my respects to your elders past, present, and emerging. And this week is NAIDOC week, so I would encourage everybody to take some time this week to learn something about the country upon which you live and or work. I know I still have lots to learn. I'd also like to take this opportunity to draw your attention to Deadly Science, which is an organisation run by Corey Tutt, who's a proud Kamilaroi man who works to provide science resources, books and reading materials to remote schools in Australia. So you can support this project by buying a cool Deadly Science t-shirt like this one. They're $30 and all proceeds cover the education of children in remote communities and First Nations kids all over Australia. Okay, so it's widely acknowledged that parents have an enormous impact on their children's physical, mental, and emotional development, with the overwhelming majority of parenthood studies focusing on parental factors that impact their child's well-being or development. But we now know that children influence their parents' development with structural and functional brain changes in early parenthood. Our understanding of the parental brain is still in its infancy, with only a handful of studies to date. And these studies vary in the time that they test, so from pregnancy up until 2 years postpartum, showing a pattern of grey matter adaptation that is dynamic and stage specific. So Hoxema and friends showed decreases in grey matter thickness across pregnancy, particularly in networks of social cognition and theory of mind. Whereas Kim and friends find increased grey matter in the early postpartum period. And grey matter adaptation was also found in fathers, with some areas increasing and some decreasing across the postpartum months. Now, these studies suggest that early parenthood involves a new neural reorganization containing periods of alternating cortical thinning and thickening in areas involved in social cognition. Throughout all of these studies, the structural changes are supporting a successful behavioural adaptation to parenthood. So in every study, the degree of structural adaptation was associated with measures of positive parental care. So mothers who experience these brain changes to a greater degree experience better attachment, more positive feelings towards their babies, and increased maternal self-efficacy, which is a confidence in one's own parenting ability. And fathers have greater intrusiveness, so this sort of rough and tumble play, which is a sensitive parenting behavior for fathers. Until now, the time course of these profound changes that appear to be quite important for our maternal behaviors is completely unknown, and this left us with some important questions to tackle. So are these brain changes permanent? Do they endure throughout the lifespan, or do they revert back to some pre-pregnancy state? And what happens if you have more than one child? So it is clear from the rodent literature that reproductive experience confers benefits that are not specific to infant care. Rats show permanent improvements in spatial learning and memory, in foraging and predatory behaviors, and increased levels of boldness, with mothers being able to sustain stressful environments for longer than non-mothers. And these cognitive changes are not unique to biological mothers. So even the mere presence of pups is sufficient to boost spatial memory ability in female virgin rats, suggesting that non-biological mothers and fathers also develop improved cognitive abilities from the environmental experience of parenthood. In addition to permanent cognitive improvements, older mother rats also show reduced amyloid deposits in the hippocampus compared to virgin rats the same age, suggesting that motherhood might be neuroprotective for rodent mothers. So our research has taken the first steps to addressing these questions by investigating the structure and function of the human maternal brain in late life. We're curious about what motherhood means for brain structure, brain function, and cognition as human women age. Reflecting this, I'm going to walk you through two studies. The first examines the structure of the aging parental brain and the implications for cognition in late life. And the second looks into the function of the late life parental brain. So both of these studies got up this year, yay, in PLOS ONE and cerebral cortex respectively. So you can check them out there if you want. So we were lucky enough to have access to the ASPRI data, which is a large cohort of elderly men and women. And we extracted cortical thickness values for 70 brain regions using T1 and T2 MRI data. We also had how many children each participant had. Analysing males and females separately, we compared cortical thickness between parents and non-parents and correlated cortical thickness with the number of children parented. And I'll take you through our coolest findings. So compared to non-mothers, mothers to one child showed thinner grey matter in the dorsolateral prefrontal cortex. The DLPFC is widely acknowledged to play a role in executive functioning and cognitive control, and is implicated in almost every study of the maternal brain, showing structural and functional changes across early parenthood. So suggesting changes to executive functioning might occur with motherhood and that these might last a lifetime. Compared with non-fathers, fathers to one child had thinner grey matter in the anterior cingulate cortex, which also shows structural and functional changes in studies of early parenthood. The anterior cingulate plays a role in social cognition, including empathy and emotional regulation, important for sensitive parenting behaviour. So the cortical thickness of the right parahippocampal gyrus, an area associated with memory performance, was positively correlated with the number of children in elderly mothers. Now this means that the parahippocampal gyrus increased in thickness with increasing number of children parented. These findings are particularly exciting when interpreted alongside our cognitive results. So we found a positive correlation with memory performance, where scores on the Hopkins Verbal Learning Test, or HVLT, increased with additional children. This finding suggests there may be cognitive benefits associated with parenthood in the aging human brain. So taken together, we show patterns of grey matter thinning and thickening associated with parenthood in late life. Now, this could reflect the pattern of grey matter changes that have stabilised following the early postpartum period, or this pattern could describe the areas that are most relevant to late parenthood, so caring for adult children and potentially grandchildren. Now, results in both males and females lead us to argue that these differences are due to the enduring environmental changes associated with parenthood. So while we were working on these papers, 3 studies came out of the UK Biobank data looking at the relationship between number of children parented and brain age in middle life, showing that parents with more children had younger looking brains in midlife. So taken together, the results of our research alongside these 3 studies suggest a lifelong effect of parenthood on the human brain, which at least in mothers may be neuroprotective in aging. So on to study 2, brain function in late life parenthood. So we used the ASPRI data again for this study, this time calculating the functional connectivity for 82 regions or nodes. We examined the relationship between functional connectivity and the number of children parented. Now, there have been many studies investigating functional brain changes in relation to parenthood. The majority of these studies have used fMRI to examine parents' brain responses to infant pictures, movies, or sounds of infant cries. These studies have predominantly examined first-time parents, usually mothers, in early parenthood, so the first few years postpartum. Very little is known about the cumulative hormonal and environmental impact of having multiple children, or about parental brain function outside the context of responses to infant stimuli and the endurance of these effects across the lifespan. So functional connectivity is the correlation in bold signal between 2 regions over time. If the activation of 2 regions is similar, so activating and deactivating together, then we can say that they're functionally connected. We say they're part of the same network or involved in the same task. And resting state functional connectivity is the same, but when the brain is not engaged in a specific task. So with resting state, we get an idea of the brain's intrinsic connectivity from a baseline state. Any change in resting state functional connectivity in parenthood would suggest that parenting is associated with global, generalized changes beyond responses to infant stimuli. So knowing that parenthood shows some benefits for brain structure and cognition in aging, we expected that parenthood might be beneficial for brain function as well. But how should we test that? So we looked at 3 established patterns of age-related functional brain change, with the idea that if we see less of these patterns, that would be indicative of younger brain activity in parents. So I'm going to give you a little crash course in 3 models of age-related functional change. So the first one is the Harold model, or hemispheric asymmetry reductions in older adults. Now, in young people, the brain is lateralized, so tasks differentially activate the left or right hemisphere. And what we see as we age and as we experience age-related decline is less lateralization of the hemispheres. So one hemisphere will recruit the other as compensation. So in functional connectivity, we would define this as increased connectivity between the hemispheres, so right to left and left to right. The second model is the Parser model, so posterior to anterior shift in aging. Parser effects again reflect compensatory recruitment of additional regions to support behavioral and cognitive function. So in age-related decline, there is increased frontal lobe compensation. And in functional connectivity, we would define this as increased connectivity between the frontal and posterior brain regions. In resting state fMRI, older adults showed decreased network segregation, or higher between network connectivity compared to younger adults. In functional connectivity, we would define decreased network segregation as increased connectivity between nodes of different functional brain networks. So we have our 3 models of age-related functional changes, which means we have 3 patterns that we expect to see in parenthood, where the opposite pattern, so decreased connectivity between these regions, would indicate resilient aging. So, using the NBS with a cluster forming threshold of 0.01, what we found was a connectome related to motherhood with 47 edges or connections, which is significant at a family-wise error-corrected p-value of 0.01. As you can see by the blueness of this figure, all of the edges in our significant connectome are negative. That is, as the number of children increase, functional connectivity between regions is decreasing for mothers. And there was no significant connectome for the relationship between number of children and functional connectivity for fathers. So when we compare the spatial distribution of our results to the patterns of age-related decline we see in the literature, we can see that the majority of our results, so 70%, fall between hemispheres, showing a disconnection between the left and right hemispheres. In other words, mothers with more children have retained a hemispheric asymmetry, or this lateralization. This pattern is opposite to what we would expect in aging according to the Harold model, and is indicative of resilient brain function in mothers with more children. We also see disconnection between the frontal and posterior brain regions, again opposite to what we would expect in aging according to the Parser model, again indicative of resilient brain function in mothers with more children. When we categorize each region according to functional brain networks, we can investigate network segregation, so decreased connectivity between networks. As the number of children increased, mothers had increased network segregation, with 96% of the negatively weighted edges falling between networks, so this off diagonal. Again, these patterns are in the opposite direction to what we see in age-related decline, consistent with a more flexible and resilient late life maternal brain. This network segregation was not evenly distributed across the brain, and some networks showing more segregation than others. So I'd like to draw your attention to the really interesting ones. So compared to what we would expect by chance, there were almost 9 times more edges between the dorsal attention network and the frontoparietal network, and more than twice as many edges between the default mode network and the frontoparietal and dorsal attention networks. So the frontoparietal network plays a central role in executive control and adaptive behavior via flexible top-down modulation of other brain regions. The dorsal attention network is involved in attention, and the default mode network is involved in introspection and self-referential processing. Now, the task-positive dorsal attention network and the task-negative default mode network are anti-correlated, meaning that when one is active, the other is deactivated. And this competitive relationship impacts behavior and supports cognitive functioning. So a segregation between these networks in mothers is preserving this anti-correlation and the cognitive functioning it supports. So more similar to the patterns of activation we would see in younger people. The other thing to note is that the frontoparietal network is flexibly co-activated with both the default mode network and the dorsal attention network, supporting attention or introspection depending on task demands. So this could mean that motherhood alters this ability to modulate between task positive and task negative processes. So segregation of these networks at rest is the opposite to what we would expect in age-related decline, and again suggests that motherhood is related to functional neuroprotection in late life. So the patterns of functional connectivity shown in older mothers were in the opposite direction to 3 separate patterns of age-related functional brain changes. This finding shows that patterns of resilient brain function in the aging maternal brain across hemispheres, lobes, and functional networks, consistent with a neuroprotective effect of motherhood on the aging human maternal brain. This interpretation is in line with previous literature in both humans and rodents, and paints a consistent picture of motherhood-related resilience in human aging, and suggests functional effects in the human brain that are both lifelong and beneficial. So we attribute these neuroprotective effects to the ongoing environmental demands of motherhood. which change and develop with the growing needs and demands of the developing children. And we know from studies in both people and in rodents that experiencing a more complex and novel environment results in a brain that is healthier, more flexible, and more resilient to aging. So it seems that complexity is good for the brain. And I think we can all agree that parenthood increases environmental complexity quite a bit. It's still far too soon to say that motherhood is outright beneficial for the aging human brain. However, our findings taken together with the current literature suggest that motherhood reshapes the brain. These changes last a lifetime and are beneficial for brain structure, function, and cognition in late life. I'll just end with a shameless plug for my current study, which is NAPPY, which is looking at the neural adaptations of the postpartum year. I'm just at the end of scanning now, thanks to COVID, and I'm in need of a couple more brains. So I'm recruiting first-time mothers 1 year postpartum and women who have never been pregnant to compare their brains structurally and functionally, see whether any differences are related to maternal factors, including cognition and mental health. So if you're interested in getting involved or you know someone who might be, feel free to get in touch. I'd like to thank all these lovely people for their support and guidance, the ASPRI group, and acknowledge our funding sources. Thank you all for listening, and I'll be happy to take any questions.
Well, thank you very much, Winnie. That was a fantastic presentation. And we have a question, which I'll put to you in a moment. But I guess preface it by saying that, of course, it is always tricky, isn't it, when we're looking at ageing, to be definitive about the likely causes of changes, given that it's an attribute. And I think that's what's motivating this question. So I'll put it to you now. So
Sure.
Given that 85% of people become parents and that having children is normative, it's arguably probable that not having children is associated, at least somewhat, with poorer health or wealth. And being richer also lets people have more children than those with less money. What then suggests that your findings are probably due to a causal effect of parenthood rather than having children correlates with having a healthier brain to begin with?
Right. I think there's a couple of things to unpack here. So first, you're right, having children is normative. It is the normal or default way, particularly for our older generations. I think it's important that we are studying parenthood on this normative process, because we really don't know how parenthood is affecting the aging trajectory in general, and particularly when we're looking at things that we know from the rodent literature. Much of what we know about aging is from virgin rats. They don't actually include rodents that have had children in their studies. So when we are looking at normative aging, we should also be factoring in parenthood and what that is influencing. In terms of associations with more children with health and wealth, it's very difficult. Particularly, you're sort of suggesting that more richer or more higher socioeconomic status people would have more children. And that's not always what we see in the literature. And I think you raise a good point. This is correlational research, and so it's difficult to, draw a causal inference from it. So we need to sort of stay in our lane in that regard. It's very difficult to say that having more children is causing a healthier brain. So yeah, we do need to be careful with that. Yeah.
Okay, I guess Winnie, that in this situation, like many others, I suppose you could potentially take some of these other factors into account, couldn't you, when you're examining differences? And if you use socioeconomic status, for instance, as an additional explanatory variable, that would help at least, address some of these questions directly.
Yeah, you're right. It absolutely would. Unfortunately for this study, the data that we have didn't include all of this extra information, because there's data acquired for different purposes, we only have the number of children that each participant parented. So we'd love to know things about, attachment, the time spent in care, all sorts of hormonal information, and indeed, information about socioeconomic status and health and wealth and things in general. We did control for education, blood pressure, BMI, cholesterol, and all of the bits that we could. So we're hoping that we're showing the best results possible with the data. And given that it's quite a large cohort and that a result is still coming out without having controlled for everything, in some ways it almost strengthens it, because the result is still there without having controlled for potentially important variables.
Okay, and I guess I guess I have a question too. And there's thanks from Ariel for your response, which cool study, no doubt.
Thanks.
I just have one observation that you might like to comment on, and because ultimately you suggested that perhaps it's the complexities of life with children that could contribute to the findings that you uncovered, and I was wondering, given that aging is a varied experience for all of us, are there other strains of information that would point to complexity more generally as a likely associate of positive aging? Are other things that complicate our lives also associated with similar findings to your own?
Yeah, for sure. We definitely see with people's working lives and the jobs that they have, that more complicated or harder jobs are associated with better cognitive reserve in aging as well. So it's not just saying this is the only way to have a more complex life or this sort of environmental enrichment. It's just potentially one of the ways that we can stay on our toes into late life.
Okay, so you're not necessarily advocating that everyone should race out and get pregnant.
Everybody go have more kids, yeah.
To keep their brains in good shape. But it could be one of a number of factors that are potentially representing a common influencer, which is effectively this notion of doing more in a way is better than not.
Yeah.
Okay.
Yeah, I think it's a gut case of use it or lose it. You just have to be more on for longer, maybe.
Okay, but this is clearly a very good start, and you must feel very encouraged to persist in this line of inquiry.
Yeah, I'm excited. I'm excited what we find with the NAPPY study as well. It's cool.
Okay,
it's
all
really
right.
cool.
Well, then I'll just congratulate you on hitting the cool study, and given that we've now reached 1 o'clock and I can't see any further questions for you, Winnie, we might conclude at that junction. Just remains for me to thank you very much. Thank you, Winnie.
Thank you. Thanks, Michael.
And that concludes our webinar for today. Thank you very much for attending, everyone. Goodbye.